Hello students, welcome to BioWhisper with Manas. Today we will continue with the second lecture of sexual reproduction in flowering plants. So let's start from where we had left, pollen grains. So there are certain advantages of pollen grains. What are those? We know that these are rich in terms of nutrients. As a result of that, they are used as food supplements. Food supplements. The athletes and the race horses. The athletes and the race horses which are used in race. So these are given, pollen grains are given to enhance their performance, to increase their performance. So what are the advantages here? Since pollen grains are rich in nutrients, they can be used as food supplements. The athletes and the race horses use them, use the pollen grains to increase their performance. Now, they can be used in plant breeding programs. They can be stored in at 196 degrees centigrade in the liquefied nitrogen form for years, and this can be used for plant breeding programs. Now there are certain disadvantages too. What are the disadvantages? This cause allergy. They cause also bronchial affliction, and as a matter of fact, parthenium. Let us know about that. What is parthenium or carrot grass? It's a contaminant that causes allergy, and it came to India with imported wheat. So there are certain disadvantages of pollen grains too. They cause allergy, bronchial afflictions. The parthenium of the carrot grass is a contaminant which causes allergy, and it came to India with imported wheat. Now let us focus on another topic, viability or the lasting duration of pollen grains. The rice, certain cereals such as rice, wheat, the pollen grains here last up to 30 minutes only. In certain families of plants such as Rosaceae, Solanaceae, and Leguminosae, these last for months, for long durations. So these were the certain things about pollen grains. Now let us move towards our other topic. What is the other topic? We have discussed about male reproductive system. Now let us discuss about female reproductive system. It consists of female reproductive system consists of pistil or carpel. Second, it consists of megasporogenesis. The third one is embryo sac, which is known as female gametophyte. That's the structure in which the female gametes are formed. That's embryo sac for you. So we have to study what pistil or carpel, megasporogenesis, and embryo sac, that is the female gametophyte. So first of all, what is the female reproductive hole? It is known as, tell me, what is it known as? It is known as gynoecium. What is it known as? It is known as gynoecium. So gynoecium is the female reproductive hole. It's the group of pistil or carpel. So gynoecium, it may consist of one pistil. If it consists of one pistil, what is it known? It is known as monocarpellary. And if it consists of more than one pistil or carpel, it is known as multicarpellary. It means one carpel or pistil more than one carpel or pistil. So multicarpellary ones are of two types. What are they? These are syncarpous, and the second one is apocarpous. Syncarpous, as the name suggests, what do we find here? Let's draw the figure here. Okay. What do we find here? Here, the carpels are fused. They are not free. And the example is papaver and hibiscus. Apocarpus means here the pistils are not fused. Yes, the carpels are not fused in this manner. These are free, not fused. Not drawn. This one in this manner. Here, here, again here. These are free ones. Example of Apocarpus, it's Michelia. Example of Syncarpus, Papaver, Hibiscus. So we have discussed about Ganoecium. These are of two types. Those have one carpel, it's known as monocarpellary. Those have which have more than one carpel, these are known as multicarpellary. The examples of monocarpellary, what are those? These are PM, that's P, mango. Examples of suppose that multicarpellary, this means though it has two carpels and it is of two types. Those which are in cynic with the those which fuse with each other. Here the carpels fuse with each other, so it is known as syncarpus, and the example here is papaver and hibiscus. Here apocarpus, that means all the sepals. Here sepals I am calling carpels here are free. Carpels of pistil are free, and the example is michelia. Now let us move a bit forward. Now we had discussed about the whole ganoecium, which is composed of pistil or carpel. So let's draw a figure first. Here we find the stigma, the landing platform for pollen grains. This is the cylinder part, the style, and here we have the burst part, ovary. We have discussed about this in the former lecture. Now let us enlarge the whole thing. Okay. It's the style, sorry, it's the stigma. Then we have the style, and here's the ovary. Inside the ovary, the empty part is known as ovarian cavity or locule. Inside the ovary, we have the ovule. It consists of, here, draw this one. Now we have the integuments. Here, let make this one first. Okay, inside we have this part. Now make the integuments from each side. Then it's the place a spot here, a stalk like a structure here. Now let us name them. What is it? So this one, the whole part is known as ovule here, and is the example of orthotropous ovule. In this orthotropous ovule, first let us know what is this. This is the outer integument covering. This is the inner integument or covering. Here we have the nucellus. Inside the inner integument we have the nucellus. And inside the nucellus we have the embryo-sac. This one is embryo-sac. So the figure here is not up to the mark, but let's get with it. So this one is stigma, the landing platform. This trial, the center part, the burst part here is ovary. Inside the ovary we have the ovule. This is the outer covering, outer integument. This is the inner integument. This is the nucellus, and inside the nucellus we have the embryo sac. Now here we have a stalk here. This stalk is known as funicle, and here it gets attached to placenta. And it gets attached to the ovule, and this point is known as hilum. Clear? So we have here stigma, then a style, outer integument, inner integument, then we have the nucellus. Inside the nucellus we have the embryo sac, and this particular part on which the ovule rests is known as hilum. This is the stalk which adjoins hilum to the placenta. This is the picture of orthotropous ovule. So I have gone through the orthotropous ovule for you so that you can understand the whole thing in a simple manner. Now let us move towards the anatropous ovule, which is given in your book. The simple is, the figure is like this. Let's draw something. First make this one, then this one. After that the integuments, this one, and we have the integuments here and also here. So these are the integuments, and these are joined by here. A funicle is here. A funicle, not in this manner. I am not getting the figure right here. The funicle is here in this manner. How is it? So let's go with it. It's here in this manner. Then it goes this way, and then it gets straight up. So that's funicle for you. This is the micropylar end. This is the chalejal end. Okay. And here we have the outer integument, inner integument, integument. Now we have the nucellus here. Inside the integument, nucellus. Inside the nucellus we have the embryo sac. So this is the example of anatropous ovule. Clear? Now let us move forward. Let us know about ovule. There are some facts attached to ovule. Then we will move towards embryo cell. Where is ovule present? It is present inside. We have seen that ovary, or can say ovarian cavity or locule. The ovules may be one in number or more than one in number. Okay. So the plants which have one ovule, the example is paddy. Then we have the wheat and mango. Paddy, wheat, mango. More than one, the best example is papaya, watermelon. These are the ovules which turn into seeds. Orchid. Have you seen papaya? You might have seen so many seeds. So it's the ovule which afterwards gets transformed into seeds. So ovule may be one. The example are paddy, wheat, mango. An ovule may have more than ovule, maybe more than one in number. Then it will. The example here is papaya, watermelon, and orchid. Now let us move forward. So we have heard about the ovule. The ovule is attached to the placenta, the place where it gets attached to this one is the funicle, the stalk. This is placenta, and the place where the funicle and the ovule meet, it's known as hilum. Clear? Now we find the opening of ovule at the micropylar end. We find the opening of the ovule at the micropylar end here where the integuments were not present. Here is the opening. It is known as the micropylar end. Now we had seen about three integuments here. This one was the integument. First it's a nucellus here. Then we had the two outer inner integument. Then we had the outer integument, right? So this is the nucellus. Inside the nucellus, right, the embryo sac. So this portion now is very important. Inside the nucellus, we have reserve food material. What do you find in the nucellus? Reserve food material for the growing embryo. And the female gametophyte, embryo sac, here this one embryo sac, it lies within the nucellus. Now let us recapitulate the whole thing. What is funicle? It's the stalk. The junction at which the funicle and ovule meet, that junction is known as hilum. junction of ovule and funicle. The nucellus here is nutritive tissue and integuments are protective layers. So this one keep in mind. This is the embryo sac and this is the nucellus. So inside the nucellus, this here is here. We have microspore, megaspore mother cell, megaspore mother cell. So now, so we have the nucellus here. Inside the nucellus we have the embryo sac. Okay. Here we have the microspore, many cells here of similar size, and one of them functions as megaspore, mother cell, MMC. Now this megaspore mother cell, MMC, it undergoes meiosis. It undergoes meiosis and forms four megaspores. Three of them degenerate. Only one remains functional. So MMC undergoes meiosis and as a result of that four megaspores are formed. Three of them degenerate, are destroyed, only one remains functional. So the formation of embryo sac from one functional megaspore. So the formation of embryo sac from one functional, your single, you can say single functional megaspore is known as monosporic development. There, let's recapitulate the whole thing. So we have the nucellus here. Inside the nucellus we have MMC megaspore mother cell. Megaspore mother cell undergoes meiosis and forms 4 megaspores. So the formation of megaspores from MMC through meiosis is known as Megasporogenesis. Clear? Now what happens? Among the 4 megaspores, only one remains functional. 3 of them degenerate. 3 of them degenerate. Okay. Now this functional megaspore helps in the formation of embryo sac. So the formation of embryo sac from one functional megaspore or single functional megaspore is called monosporic development. Okay, let's move now. Now, how does this monosporic development take place? How does this monosporic development take place? At first, the functional megaspore undergoes mitosis. Now, how many here? 2 nuclei are formed within the same cell. Then we have 4 nuclei. At last we have 8 of them. 2, 3, 4, 1, 2, 3, 4. So these divisions are strictly free nuclei. Here no walls are there. These developments are free nuclei. But afterwards cell formation takes place. How does it take place? Here, Here we have the 3 cells here. And now these cells are covered by wall. Here we have 2 nuclei and we have the central cell here. Here we have the egg apparatus. This is the synergids, and here we have the finger-like structure, which is known as filiform apparatus. Filiform apparatus, finger-like structure. These help the pollen tube, or these guide the pollen tube to move through the synergids. Synergids are the paths which leads the pollen tube towards the egg cell. This one is egg cell, and these two are known as synergids. So the egg cell and synergids, they are commonly known as egg apparatus. Okay, this one is the central cell, and we have 2 polar nuclei here. 2 polar nuclei, and these are 3 of them are antipodal cells. This is the chalazal end, and this one is known as the micropylar end. So here we have three nucleus here, and now they are surrounded by valve 2, 3. Here inside we have two nucleus, the polar nuclei. It is also surrounded by a valve. That is known as central cell. And here we have the two synergids and the egg apparatus. Two synergids and here this egg cell. It's the egg cell, and the whole thing is known as the synergids and egg cell. These are known as egg apparatus. And here we have the finger-like structure, which is known as filiform apparatus. It guides the pollen tube to through these synergids. It can move through any of the synergids, and this is where the pollen is supposed to meet the egg cell. I hope you have understood the whole thing. Now, how many nucleus do we have here? How many nucleus do we have here? 1, 2, 3, 4, 5, 6, 7, and 8. So we have 8 nucleus. How many cells are there? 1, 2, 3. And here we have the fourth one. Inside there are two nuclei, but the cell is central cell is first one. And here we have 1, 2, 3. So 3, 1, 4, 4, 3, 7. 7 cells. So an embryo sac is a 7-celled, 8-nucleate structure, and it is known as now we have the 7-celled, 8-nucleate structure of embryo sac. One, two, three. They have the cell here, this one, and we have the egg apparatus here, and this is the filiform apparatus. So this is known as embryo sac, and it's the female gametophyte. Where female gametes are made. So till now we have studied about the female reproductive organs and how does megasporogenesis take place and how embryo sac is formed. We will continue with this topic in the third lecture. Until now, if you haven't subscribed my channel, do it now and do love my videos. Thank you.